So I guess I've been already introduced. For those that don't know me, I'm working in Elsa's group, and I'm a programmer working on tools for the taxonomic analysis of shotgun metagenome samples. We are developing different tools in our group, and I'm the main developer of one of those tools. And I will focus on this tool, but I will also give you a short walkthrough, and I will show you what our other tool does and how it works very quickly. So, a quick reminder, I guess, from the previous talks, you already guessed how it works. So yeah, you couldn't kind of divide the steps in shotgun metagenomics in two parts. One is more sequencing related, so it's technical, it's experimental. You take a sample from a natural environment, you sequence it without any culturing steps in between, and then you after sequencing you have the primary sequencing products, which are reads, and you can also assemble these reads into longer contexts. But no matter what you do, you will the input to the downstream analysis steps will be either reads or context. So to answer more ecology-related questions, what people usually do is they first quantify the abundances of the different operational taxonomic units or taxa in the sample, which we term taxonomic profiling. That's just the first step, actually. The second step would be the binning. And in taxonomic binning, we try to assign every single sequence, either read or contig, to a species or high-level taxon. And of course, if you have a binning, you can infer a profiling, but you really need the binning to answer the more interesting questions, like what gene can we link to which genome or taxonomic unit. Okay, so these are the two tools. I'm the developer of TaxoTK, and I will first show you the theory behind the algorithm, and then I will give you a short intro in how the software works and how it's applied to your samples. So there's a second developer, Ike, who isn't here today, but he's also working on it. We developed the tool using C++ and Python, and we do use sequence similarity, and we try to approximate an evolutionary neighborhood for genes or arbitrary DNA sequences in order to infer a taxonomic ID or a taxon for this unknown piece of DNA. So to do any kind of analysis like that, you need to have homologs. You need to have similar sequences. So we rely, as many other tools, we rely on a sequence homology search step, where you have your unknown metagenomic sequence, either read or contig, and you search it against a reference database, and you will end up with a list of potential homologs. And we can plug in any aligner that you like, and we try to make it work with also more noisy nucleotide alignment to be fast enough. And as reference sequences, you can also use whatever you like, as long as you have nucleotide sequences in terms of FASTA files. So this is the overall workflow. We try to decompose the problem to make it better solvable. The input sequence, like read or contig, is divided into individual segments, which we find homologous regions for in the database. Actually, you infer the segmentation from the local alignments to get these chunks. And later on, for each of this set of sequence segment and homologous segments from the reference data, we apply an algorithm called the realignment placement algorithm to infer a taxonomic prediction for this tiny or long, depending on how the homology search went, piece of tiny part of the query sequence. And since we do that several times we have to come up with consensus prediction for the entire sequence, and we have a consensus algorithm that does that after this procedure. So the core part is, of course, this realignment placement algorithm, and which actually identifies the evolutionary neighbors of this query part. And in this case, we have the query segment and the query the homologous segments as input to the algorithm. And the idea here is to kind of use a phylogenetic structure that's in the data. Just imagine you would throw those sequences into a multiple sequence aligner. You would get out an aligned genes or aligned sequences, and you could construct a de novo phylogenetic tree from that. And using that tree, or you could root that tree and put it into a format like that, where you have 3 subtrees containing homologs, one that is most similar to the sequences, to the query sequence, and one that's most distant, and one that's in between. You can root the tree because you know the taxonomy. So you find that each of the subtrees represents a monophyletic group, and that's the definition of that's a modern definition of a clade. And so if you use the group members of a monophyletic group, and you use that to infer a prediction taxon, you actually you make the most correct prediction that you can. So the realignment placement algorithm is just a fast algorithm to compute something like this tree on the right side, but it doesn't actually compute the tree. It just does pairwise comparisons. It does also not need any parameters. So oftentimes you set threshold parameters to infer this neighborhood, like with Megan, for example. And this is completely parameterless. So at the end, when you know these members of this monophyletic group, you can apply a lowest common ancestor algorithm to get a proper taxonomic ID for this query segment. So just to very quickly, I don't have enough time, I think. We perform 2 runs of pairwise comparisons. So we don't have to infer multiple sequence alignment, and we don't have to construct a tree. And by kind of grouping the sequences to the 2 homologs, the most similar one to the query, which is denoted as S here, and an outgroup sequence, we kind of construct a partial tree and can infer such a taxonomic prediction. So it's quite fast and applicable to metagenomic data. So I'm sorry. Maybe I should tell you why we do that. So we segment the query sequence because it's feasible. We can parallelize all these different RPA calculations. It's also more robust because we get a number of predicted taxa that we can combine. So if there's like a noisy region on the query sequence or a misassembly, then it won't impact your final results as much.
Could you identify a misassembly this way?
Yeah, we come to that. So it gives you a very robust overall procedure to assign query context or reads to a taxonomic ID. So the overall advantages are basically that you can infer also precise taxonomic prediction for novel taxa, so not ones that you only have related homologs for. And it scales very well with the length of the sequences. There's no limit on the input length of the query sequences. It can be short reads or even entire genomes. It can be noise. It can be something like PEC biodata because it kind of averages out the noise. It's parallelized, so it's fast, but it also has a good theoretical runtime because we have a linear number of comparisons. And the whole thing is kind of modular, so we have implemented everything in individual modules. On the right side, you see that for each individual bin, you have a high level of purity, or in this case, it's the precision of the bin, and very compared to other tools, that's pretty good. 90 to 100% purity is quite good, I think. But you still depend on this homology search, so you cannot get rid of the homology search by using this approach. So that's one disadvantage. So if you want to apply this software, you basically have to go through some steps, but I tried to keep it very simple. So there are 4 steps that you have to do. You have to download and unpack the software, and it's already pre-compiled, so you don't have to bother with dependencies. This is the extended version of the software, so it comes with all the aligners, all the different tools that are needed to construct the workflow. You need to download reference data, but we are providing a sample, a script that does that for you. So you can download the reference data, and then you just only have to decide for an aligner that you support out of the box and create the aligner index. And then you have to run the pipeline. So suppose I could log on to the internet, which I cannot. I will try to do it locally. So when you unpack the software, you basically end up with a folder that looks like that. So it does have some subfolders, documentation, a binary folder with all the binaries inside. We do provide 2 sample workflows that you can apply, and the scripts to get started, to download the ref data, to create the indices, and so on. So suppose you want to classify something, you need to fill in this. You need to have the data ready. So here in the ref data, we do have some data, and it's pretty easy. It's just a FASTA file, the taxonomy version, and the mapping between those 2. So you don't need to prepare any special files. The aligner index, it depends on the aligner, but it looks pretty much the same. You want to go ahead and classify data. Let's do that. There is a packed bioreads file. It's reads between 1,000 and 3,000 base pairs long. It's 33 megabytes large, so we try to classify that on the laptop, including the alignment step. So we just call the pipeline script using BLAST, because it runs on my laptop. We give it the FASTA file and try to classify. Okay, that's already working. Let's wait. Okay, maybe I'll show you. So it's aligning the sequences at the same time, and then whatever sequences are produced are consumed by the Taxator toolkit, and are used to classify the sample. As they're running parallel, and yeah, that works. So we go back to the presentation. I'll quickly walk you through the pipeline. This is how the pipeline works. We have basically 3 programs. We have the aligner, which is BLAST or LAST in our workflows. We have Taxator, which applies the realignment placement algorithm. And we have the consensus algorithm implemented in the program binner. So if you want to implement that workflow, you just plug those together. There's a bash script that contains the functional workflow. If you define the variables, that's complete, that's working. That's what we're running in the background right now. And we try to keep everything modular, and we try to make it possible to recombine those parts for other kinds of applications. So the input alignment is just a separated table, which must contain the positions on the reference and the query, and as well as an alignment score. So that's what you can squeeze out of any aligner that I know of, at least. The reference and query, you just need the FASTA files. You do need taxonomy as you can download from the NCBI website or the FTP server. No special data is required. No special file formats as well. So this is the most interesting part. The output of the text data, which is basically the predictions for the individual sequence, query sequence segments, is written in the general feature format, and it preserves all the information that we can get out of this algorithm. So it contains more information that we are using in the consensus algorithm at the moment. So the nice thing is that you can actually go and load that into an assembly viewer, or you can load it into a genome viewer, and you will see which parts of your sequences are assigned to which taxa. Or maybe if you suspect misassembly or chimera, you can investigate in these issues. We do have C++ and Python parsers to parse that file. We're using it to parse it with Binner. The final output is just a tab separated file. You have sequence identifiers, you have the predicted taxonomic IDs, and you have a support value that you can use to quantify how reliable the prediction is. So, if we're going back to where we started the program, so it's still running because it's my laptop computer, but on the server it'll probably run through in 2 or 3 minutes. We could go to the output folder, which I already calculated before. Oh no, that's not the one. That's the one. So there will be an output folder that's called like your input sequence, wherever you are. And if you go into that folder, there's a bunch of output files. The primary output, which is the final predictions in the sample.txt file. The GFF file you find here, which contains these segment predictions and very detailed output. We do preserve the alignments as well in this simple tab separated format. If you wanted to look at the summary files, which we also produce, to get an impression how your community looks like. So that means, let's see. Maybe I should stop the computation. Okay. Here we go. So you do get something like a taxonomic profile. These are accumulated support values. And this is kind of your community that you find. And you'll see that not everything is classified down to the species level, simply because of the phylogenetic structure of each individual input sequence. So sequences can be ambiguous. They can carry not a sufficient phylogenetic signal, so they get classified to higher levels. You could put that into a picture. Like we plan to integrate Corona in the next version, so you can get the same information in an interactive pie chart, and then you see better how it distributes across the different taxa. You can search it and click on it and so on. So yeah, that's just the output. That's just the profiling output. And you can of course look for genes in the output and in the files and so on. Okay. So I'm going to the second short part, which is about the PyThiaS Plus software. It's developed by Ivan Gregor. It's written in Python and C. And this is a very, very quick introduction. So just as a reminder, the composition-based classification approach works the following. You do have training sequences or input reference sequences as you have with Taxator TK, with similarity-based methods. And you're using this data, or more precisely, you're using k-mer frequency features derived from that data, together with the taxonomic annotation to derive a model, a static model. And later on, you can use that model to classify each individual query sequence and infer a query taxon for each of the sequences. So it's a multi-class classification thing, and your taxa are your classes, and there's a general issue that the more classes you include in your model, the less appropriate will it be for your sample if many of these classes are not relevant for your sample. So that's the general idea. The plus extension of Philopithia-S basically adds this a prior part, which uses a marker gene analysis step to infer the community composition, and it also allows to accumulate additional training data for the model. And that training data, together with the information about the community members, the number and the type of the community members, is used to then train a better model that is specific to your sample. And using this model, you can do much better than using a general model. Yeah, it's not much. Basically, you don't need human intervention anymore. So the pros is for this composition-based approach are that it's quite fast. So it's not like an online calculation algorithm as with Taxator-TK. It's linear in the input in terms of base pairs, and it has to have a higher recall. So it classifies more on levels like species, genders, and family. As you can see on the right side, the blue bars are much larger, but you also get more errors. So it's a slightly reduced recall, but you are able to classify more data. And it's particularly good for the major players of your community. So the abundant species can be recovered quite well using this composition-based approach. And you get reasonable precision, as I already said. The major drawback is that you need a stable signal, a stable compositional signal, and that you only get with sequences longer than 1,000 base pairs usually. So you cannot use it to classify raw reads at the moment. I don't know about backbio reads. And it might also miss some of the rarer taxa because it's kind of, it constructs a more limited model and uses that to classify the data. So I'd like to acknowledge, of course, my PI and the other developers, but I'd also like to mention all the open source developers that are working on the frameworks that we are using, in particular that I'm using, like SecArm Boost and 3D.hh. And if you're interested in the theory, or basically the preprints and how to get the software, these are the addresses that you need to know. Yeah, this is the end. Thanks for listening. I'm glad to take your questions, if there are.
From the marker gene analysis, you get information both about who's there, but also how abundant each member is. Are you able to use that when you do the training?
You don't use the abundance at the moment. I mean, you are looking for additional training data, and you want to construct like the number of classes in your model. So we're using these informations, but we're not using abundances from marker genes.
I can imagine, like in a Bayesian setting, if you knew the abundances of each group, you'd have some prior probability that any read would go to that particular group.
Yeah, that's probably that's true.
Yeah,
but,
it's a nice idea. But I think the current markers are not that many. So
yeah, the protein markers are sparse, right? Because the genome sequence databases are.
Yeah, and then you don't know how copy numbers play into that. And
should all be single copy.
Okay.
No, but it makes sense also because the amount of training data you put in sort of impact how frequently something is assigned to a particular class, even if you don't make it a Bayesian setting, sort of makes a difference. So if you have more data, I think that would be good.
Yeah. So yeah, I think there are ways of doing that where you don't need to worry about any of these things.